one-on-one conversation can be going on.
सर पानीगिरी सर चले गए।
हाँ वो डिस्कनेक्ट हो गए हैं, वापिस अभी रिजॉइन हो जाएंगे। लाइव।
Okay, I am sorry for the inconvenience. Okay, let me share.
Yes, 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 sir.
Okay, so today we will discuss about one of the important topic, as sir has mentioned, that is artificial seed or synthetic seeds. So while you are discussing about any type of seeds, first of all, you should know about that what might be the different things you can have with this seeds form. Seeds include the outermost layer, that is testa, outermost layer testa, and inner to it testa there is a layer called as tegmen, or we can call them together as seed coat. We can call it as seed coat. Okay, and inside it there will some seeds do not possess endosperm, some seeds possess endosperm. So those seeds which possess endosperm are called as endospermic seeds. Which are not, they are considered as non-endospermic seeds. Actually, this endosperm provides the nutrition, or we can call it as the nutrients availability to the developing seed required or furnished by endosperm. Okay, so these things, that is the generalized way to define a seed. Okay, but in case of we are using this terminology, one is artificial or we can call it as synthetic. Why it is being made? Do you think that this will be manufactured in any of the factory? Not such like that, but one of the biological factory, that is in vitro or plant tissue culture technology, by means of which we can prepare, we can able to synthesize or made this type of seeds. So if you discuss about this, let us discuss its definition first. Definition is, I have mentioned few of the points one by one. Okay, and how question may ask from it, I will tell you. So first of all thing, the artificially we have to manufacture. What are the terminology? Encapsulated. Encapsulated means which one we are going to capsulate, that is somatic embryo. I am coming to this word later on. Why we can call it as somatic embryo. So artificially encapsulated somatic embryos. What might be the other features? The other feature will be you can take shoot pods. If you encapsulate it, that can also be considered as artificial seed. Along with it, that you can take cell aggregates, or in tissue culture we can call it as callus, okay, or any other tissues of the plant parts. that can be used for sowing as seed. That means the normal condition, we are taking the seeds and put them into the soil and it will be germinated. Likewise, this can be also sowing as seed. That's why we can call it as artificial seed. And the most important thing is that that can be grown in both the way. How? Either in vitro. In vitro means inside the lab condition, or we can call it a inside the test tube or sugar tube rather. Okay, and outside the lab condition, that is ex vitro condition. They have that ability to this, and naturally they have the potential for storage. So all these definitions part or synthetic seeds are important. Question may ask, among the following, which one can be considered as artificial seed or synthetic seeds? Then any of the part can be put as option. Okay. So accordingly, you have to remember. Next one is somatic embryo. Why we can call it a somatic embryo? Why not zygotic embryo? Here I am writing two words. One is zygotic, another one is somatic. Can anyone differentiate these two terms? Zygotic embryo and somatic embryo. Zygotic, what is the meaning of zygotic? Or what is the meaning of somatic? Yes, anyone try?
Somatic means vegetative cells,
Very good. What about zygotic?
reproductive cells.
Yes. When there will be fusion of male and female gametes occur, then it leads to zygote. Okay. And from that zygote, if embryo is being derived, we can call it as zygote. Okay. Whereas somatic means any plant parts other than the reproductive parts can be taken for the manufacture of embryo. Let us say if you are going for the callus production, then this callus one can lead to embryo. Okay, embryo has the capability or it has the remembrance that which plant it is going to produce. Okay, so from any of the vegetative parts, if any embryo is being produced, then we can call it as somatic embryo. Got it? So we can take also shoot pods of any of the plant, cell aggregates of that one. Okay. Likewise, these are the major. Another thing I would like to add over here that there is no need of hardening. There is no need of hardening. Have you heard about this? Perhaps you must have heard about this word yesterday class. What is hardening? Micropropagation में कल पढ़े होंगे. Hardening. This is also called as transplantation. What is that? In micropropagation, the last step of micropropagation is hardening or transplantation. That means while you are trying to making a plant in the tissue culture lab, there will be regeneration of roots and shoots occur. A total plant will be occur. After manufacturing that, you have to transplant them into a polythene bag and you can transfer it to greenhouse condition. Later on, you can switch it over to the farm condition. So the last step will be transplantation. So here I have mentioned this word, no need of hardening in case of artificial seed. So this title part is very important. This definition is very important as per synthetic seed is concerned. Okay. Now let us discuss how this can be possible. I have taken this from this paper, Bose and Huck, in the year 2019, how they have tried to manufacture the artificial seed. This is that artificial coating. This grayish part is artificial coating, and this part is the explant which we have discussed. This is a somatic embryo from which you just mark there is starting of the embryo part. This is a group of artificial seeds. This has been taken an internodal part from which this regeneration can be possible. Here you mark after a few period, they are and just degrading this artificial coating, and these are the initial leaves, these are the roots production, and how this development has been done from this artificial seed, and later on becomes a full-fledged a plantlet. So you can, if you want to read, then you can follow this paper also. Okay. So how this thing happened and what are the techniques behind it, let us discuss. If we are looking at the history of it, first of all, this fellow Murashige, he first of all coined the term artificial seed. You must have heard about Murashige and Skoog medium. In case of tissue culture, in simple word, we can call it as MS medium. So this M stands for Murashige, and he is that fellow and who coined this term artificial seed in the year 1970. Okay, and he tried to propose what he proposed. He proposed that if we encapsulate the somatic embryo, then we can have the probability of making a artificial seed. And further, other scientists were tried to advance it. One of the scientists is Redenbach et al. And the other two scientists, Kiko and Jennings, they also tried to advance this technology. And this Redenbach et al., they have first of all patented this technology in the year 1980. Okay. In India, it was first of all started by the scientist Buffett. What we can call them as Buffett or Buffett, I could not say, but he started this work in Sandalwood as well as Morris Alva in the year 1980. That's why he considered as father of artificial seeds in India. Father of artificial seeds in India. So here, objective point of view, you have to remember this as well as this one. No doubt, this one patented technique by Redenbach et al. Okay. Now, what are the system of synthetics? So there are basically two systems. One is desiccated system, another one is hydrated system. Have you heard about desiccation? What is the meaning of desiccation? Can anyone? What is the meaning of desiccation? Yes. Desiccation means it is a position or it is a time period where though there is withdrawal of water occurs, or you can call it a dehydration occur, once it is being putting or recovery of water occurs, then it again survives. Then we can use the term desiccation. So this artificial seed condition, there is a system called a desiccated system. What does it suggest? It suggested that if you are taking the somatic embryo, then it should be sustain the desiccation time period. That means whatever water is being removed from it, and whenever water is available till that day, it can be hardened to that condition or accustomed to that condition, and later on, once it is being done, then it is being encapsulated by a suitable coating material. We will discuss that coating material later on. As we have seen in the last part also, the coating material, how it is looked. So under this category, the desiccated system, first what they will do, they will collect the somatic embryo, which is already developed in the plant tissue culture system, and these somatic embryos later on, it is being treated with a suitable polymer. And these polymers, that also we will discuss, these polymers, once it is being attached to this or encapsulated this somatic embryo, followed by little bit drying, so that it will not create any problem to the or infection to the somatic embryo. Or you can go for ABA. Why it has been mentioned here ABA? Why it is being utilizing ABA? In the normal seed production case, there is a phase called as dormancy. Once seeds are produced by the plant, it is followed by a dormant period. And this dormancy is being supported by one hormone, that is abscisic acid, so that it can remain for a period as it is. Okay, there is no question of further germination. For that reason, ABA can be treated during its latter part, so that it can survive for a certain period of time period, because you have to store it while you are not utilizing that for germination process. Okay, and those somatic embryos which are utilized and they can sustain this desiccation period, that's why they are said to be desiccation tolerant species. Desiccation tolerant species of plant. Not all the plants has the ability to undergo the desiccation period. For that reason, whichever plant can able to sustain this condition, then we can go for this desiccation system of artificial seed production. Okay. Here I would like to mention one word, that is resurrection plant. Resurrection plant. What is the meaning of this resurrection plant? Which possesses the desiccation tolerance capacity to withstand dehydration. Okay, as I have mentioned earlier, and recover fully upon rehydration. That's why we can use this term desiccation. Let us take few examples of this type of plant. This is one of the plant. It is named as Everlia rhodophensis. Okay. Another one is this one, that is Goya hygrometrica. These are mostly the mid-Asia region. These two plants can grow. You can Wikipedia it so that you can get more idea about these two plants. But they are coming under the resurrection plants category. So initial thing, we have to go for this desiccation tolerant system, so that we can develop the somatic embryo. Next one is, this is the how you can go for the production. This is A part, which is somatic embryos. All of them are somatic embryo. In a 8-liter bioreactor, they wanted to grow, and this has been taken from the carrot, gajar. Okay, and it is being incorporated in high osmotic media. You must have heard about osmolytes. Here I would add certain thing. Osmolytes. Plants generally prefer to produce osmolytes during the water stress condition or stress condition. You can note down certain osmoprotectants. You can write down. First one is proline. Second one is lysine betaine. Lysine, lysine betaine, BETAINE, lysine betaine. Third one, spermidine, SPERMIDINE. Fourth one, formin. Fifth one, eucricin. In some cases, mannitol. Or sorbitol. These are all osmolyticum. That means they can able to maintain the osmotic behavior of that compound, or we can say that pH can be also maintained by applying this osmotic compound. So you have to remember all of them, okay? Whenever it is asked in your exam, which one is considered as osmoprotectant or osmolyticum, then you can have idea about this. Okay, so I think all of you have noted this. Next one is, this one is after uh, the encapsulation. These are the desiccated embryos. Now next switch over to the hydrated system. Hydrated system case, certain coatings were being used, and those were called as hydrogels. Hydrogels, why it has been mentioned? Because they can easily dissolve. These gels are easily dissolved in water. This is also one of the important points as far as MCQ is concerned. Whether hydrogels are dissolved in water or not. So somatic embryos which are encapsulated with hydrogels, they can be easily dissolved in water. So what are the different hydrogels? These hydrogels you have to remember. What are these? First one is sodium alginate, second one is potassium alginate, third one is carrageenan, that is one of the algal extract. Fourth one is sodium pactate or sodium alginate with certain gelatin. So you have to remember this. How we will incorporate that, what are the different steps that we will discuss one by one. Okay. But one thing is that one problem of hydrated artificial seeds that these are very sticky in nature. So while utilizing this is difficult to handle. Okay. For that reason, once it is being utilized, it should be dry in an open air so that the further process cannot be hindered or stopped. Okay. So what you can do, either you first do this, later on you can supply a waxy coating over the beads so that there will be no connection among them. So it can be possible later. And once it is being made, after this waxy coating, you can have to plant this once it is being produced. So it won't support the more time of storage. Okay. Now come to this explant. Which explant can be taken for this hydrated system? Okay. You can go for callus culture. Callus is the best material for this, and for that reason you have to induce the callus. I think all of you know what is callus, undifferentiated mass of tissue. Okay. Or you can go for the somatic embryos in the callus. And once it is being done, then somatic embryos can be processed further. Okay. So these are the techniques by means of which you can go for it. So first you will have to choose the explant, which explant you are going to take. Then you can go for callus induction. Further callus can be induced to make somatic embryo. Later on, if it becomes, the somatic embryo can be proliferated further, and histodifferentiation if required. Histodifferentiation means is you want to take any anatomical feature for it, then you can go for it. And finally, you have to go for the encapsulation of somatic embryo by means of applying those type of gels. Okay. And later on, you have to standardize. What is the meaning of this over here? Whatever process you have done earlier, that has to be required to be standardized. That means how much hydrogels you want to pour over here, what should be the thickness of that you have to standardize for particular artificial endosperm or somatic embryo. Okay. And once it is being done by several experiments, then you can go for the mass production of those artificial seeds. Once it is being done, you can have the idea that By applying this much somatic embryo, this much hydrogels, we can able to generate huge amount of artificial seeds. So that can be possible. And later on, you can have the ability to transfer it into the field planting or greenhouse. Mostly, whatever tissue culture things were going on, so for that reason, you have to be considered the greenhouse condition. Gradual process of hardening is better for tissue culture condition because initially you have grow them at 16 to 18 degrees Celsius. Later on, you can switch it over to directly 27. So it could not able to possible. So during the encapsulation technique, that means to capsulate the somatic embryo, there are two techniques: dropping method. Another one is molding method. Let us discuss this one first. Dropping method. In tissue culture, what would happen? This is a protocol type of thing. Okay. Initially, what you will do, then later on, what you will do. That's why this is totally a protocol. So, dropping method case, first initially you have to take the somatic embryo, which is already developed in the PTC lab, dipped in hydrogel. Okay. And once it is being, let us say this is the embryo, you have put them into the hydrogels, then it will encapsulate the somatic embryo. Whatever the remaining part of it, it should be dried. Okay, it should be, and hydrogel we have already discussed. Either of them, alginate, sodium alginate, in some cases, carrageenan, which we have discussed. This sodium alginate, this alginate part can be derived from one of the seaweeds, agar. Okay. In some cases, certain seed gums were also being utilized, such as guar gum, locust bean gum. These may be also considered as the utilized for this process. Okay. So, how much, as we discussed about the standardization, it should be 2% of this solution, sodium alginate solution, and importantly, it should be taken in MS media or prepared in MS media. Why? Because MS media has that supplement or the standardized supplement so that somatic embryos can easily germinate. That nutrients can also be provided from this MS medium partially. Okay. And once it is being encapsulated, then it can be added as drops into a calcium nitrate solution. Later on, we will see that this calcium nitrate can be exchanged with the other chemical. So, what you will do? First, embryo you have taken, then encapsulated, then dip it into this calcium nitrate, or drop wise you can add this calcium nitrate solution. It may be around 100 millimolar. Then you have to keep it as such for 30 minutes, around 30 minutes. Then it will be gelled by calcium alginate, or you can incorporate into the calcium alginate solution. So this is the step-wise process. Okay. And lastly, it can be rinsed with water so that extra whatever is not there, it can be removed. And it will be totally a smooth coating can be made, and we can call it as artificial seeds, or we can call it as synthetic seeds. So this is a very easy process, nothing to worry. You can do it as practical in your lab also, easy to handle. So this technique was first of all done by Redenbaugh in one of the plant, that is alpha somatic embryos. We have discussed that Redenbaugh has patented this technique. So he first of all utilized this in case of alpha plants. It is one of the easy to handle that. Okay, instead of calcium nitrate, as I told you, you can also go for calcium chloride, and it is easily available in your lab. easily available in your lab, that is calcium chloride. Okay, so by utilizing this one, you can able to make the encapsulation process easier. Okay, or it can be treated with sodium chloride, but best one is calcium chloride along with this calcium alginate. Okay, so this is regarding that hydrated system where we can encapsulate. Next one is there is a requirement of certain adjuvant. Adjuvant means certain extra protected thing which can be required by making this artificial seed or synthetic seed, or it will provide extra protection, such as what might be utilized, certain growth regulators, okay, certain hormone type of thing. If it can be utilized with certain more protection herbicides, so that if you try to spray the herbicide in the farm, it should not hinder it, as well as you can apply insecticides to it or fungicides to it or mycorrhizae to it, so that they can able to sustain for a period. So what might be the actual function of it? This will prevent the embryo from desiccation or any of the mechanical injury. Here I would like to mention that synthetic seeds, you can note it down in your copy also. Synthetic seeds differentiate from zygotic seeds writing here short form zygotic seeds or embryo, in that synthetic seeds don't have or do not have seed coat. They are without seed. Synthetic seeds do not possess seed coat. This is one of the important questions mostly asked in synthetic seed MCQ. Okay. So for that reason, if these synthetic seeds after encapsulation, if these adjuvants were being utilized, they will supposed to prevent from desiccation or any of the mechanical injury. Mostly the zygotic embryo with this seed coat, they are the protective measures which is naturally synthesized in the plant seeds. So for that reason, these adjuvants are utmost important. Okay. Further, if you try to add some other things such as activated charcoal, so what it will do? Activated charcoal improves the conversion and vigor capacity of somatic embryo. Let us say you have made 100 seeds, 100 artificial seeds. So if you are applying this activated charcoal, maximum chances that this majority of them may grow. Majority of them grow. But I would like to mention here that there are certain disadvantages of making this artificial seed in the last slide. Okay, all these processes are very good, but success rate we will discuss in the last part. Okay, cost effective, but how much it is successful. But in which plants, which category of plants, why there is a requirement of that also we will discuss better. Here only the techniques, let us discuss first. Okay. So apart from charcoal, you can utilize some other related substances. But what might be the process why charcoal is being utilized? Because the maximum chance that whatever nutrient that somatic embryo having, inside that hydrogel that can be remaining as such. Okay, and it will slowly releases so that the growing embryo can be developed in a proper way, because growing embryo require residual nutrients so that it can initially germinate like this, later on it will be like this, later on it will be like this, later on a leaf-like structure can be generated. So residual requirement of these nutrients are there. So charcoal allows them to gradually available to the growing embryo. Okay. So this technique can be applied to all these explant category. Either it may be directly you can take the somatic embryo, or auxiliary buds, or apical buds, or adventitious buds. In all these four cases, you can go for this hydrated system, and after making this, you can apply the charcoal. So that is dropping method. Now let us move to next method, that is molding method. In case of molding, why it has been mentioned? Because it is temperature dependent. Temperature dependent gel, such as gel right and other. So it is particularly very specific and you can be utilized. So what it will do? Generally, whatever cells you are taking, it should be coated with the gel, but it should be at lowering of the temperature. Let us say, generally, initially you are going for 25 degree, but it will not like that. It should be around 16 to 17 degrees Celsius. Gradually, you have to add. Otherwise, this gel-right and agar will not work. For that reason, we can call it as molding method. Okay. So this is the gradual step, how we can go for it. Let us say you have taken carrot as your explant. Take 1 mm by or 2 mm by 2 mm part. Okay, 1 mm to 1 mm or 2 mm size. You can cut it after a proper, that is utilizing certain chemical agents. Then you can grow them in MS media, and you can go for the callus culture. Mostly 2,4-D hormone is preferable. 2,4-dichlor. Anyone know about the full form of 2,4-D? What is the full form of 2,4-D? This is a hormone. Yes, anyone try? It is 2,4-dichlorophenoxyacetic. Dichlorophenoxyacetic acid. This is one type of oxime, artificial or synthetic of dichlorophenoxyacetic acid. Naturally, natural form of 2,4-D, if you are applying to the farms, it can be considered as herbicides. Okay, it can kill, rather not herbicides, sorry, it is weedicides. It can kill the unwanted plant, weedicides. Small weedicides, it can be killed by this. So if you are applying 2,4-D, it will develop the callus. Then for more callus production, you can switch it over to suspension culture, so that more of the callus production will be there. It can be reconsidered for suspension culture for several generations. So after, let us say, 24 to 48 hours, more of the callus will be produced. So you can transfer them, or you can go for the soft culture. Again, you can go for the suspension culture. So depending upon your requirement, more of the callus can be produced. Then you have to go for transfer it into such a condition that you can have a pro-embryogenic mass production can be produced, so that the somatic embryo can be developed from this. Here in large scale production, there will be certain bioreactors where you can produce the large number of somatic embryos. Once these somatic embryos are produced, here you mark, there will be two-way mark, so that one is for the rooting site, one is for the shooting site. Then it is being encapsulated by means of any technique which we have already discussed, and later on it can be stored. And as and when required, we can transfer them into the field so that it can able to grow. So this is the cycle by means of which you can go for the somatic embryo production. Okay. Another category I would like to mention here, that artificial endosperm. Okay. Artificial endosperm, as I already mentioned, the endosperm, that is the nourishing part which nourishes the developing embryo. Okay. So for that reason, if we are providing the endosperm, artificially made endosperm to that somatic embryo part. Okay. So for that reason, if we apply these nutrients which are mostly found in the embryo part to the somatic embryo, then it can be also being enhanced during the seed production or the germination process. So if you are applying the addition of nutrients or certain growth regulators, they are considered as the artificial endosperm. So along with this somatic embryo, if we apply this artificial endosperm, that can able to grow during the germination stage. Okay. Mostly this synthetic seed stored about 6 months. Okay. And here you mark, they are without losing viability when they are stored at 4 degrees Celsius. So it is also important as per MCQ is concerned. They can able to store for 6 months. Okay, within that period, you have to go for the germination process. Now come to the what are the uses, advantages, and disadvantages. So first of all, the use among this use, if you try to make certain hybrid plants which are generally having unstable genotypes or show seed sterility. So we have to choose such type of plants where the stability of the genotypes are prone to attack. Then we have to choose that so that we can have the somatic embryo of it and we can able to produce those plants. Okay. In some cases, seed sterility is there. No doubt the seeds are produced in plants, but those are not able to germinate. So for that reason, we can take those somatic embryo from that plant and we can go for it. Okay. Second use of this one is cryopreserved artificial seed. I will discuss this part, cryopreservation, tomorrow. That is liquid nitrogen minus 96 degrees Celsius, how it is going on. So this cryopreserved artificial seeds can also go for germplasm preservation, and particularly in recalcitrant species. Recalcitrant, what is the meaning of this one? Recalcitrant means if you apply any heat to it, it cannot sustain that period. It will be killed by that one. You can Google it, recalcitrant. So it will be easier for you. This is also coming as MCQ. Okay. Third point is, what might be the role of endosperm in developing embryo that can be studied by means of somatic or synthetic seed production. As well as we can able to produce a large number of clones and particular to allied species, and that too a cheaper cost. Very less cost will be required to make this type of product. Okay. And also to supply certain adjuvant like this which we have discussed, so that can be utilized. Let us say if you have applied certain pesticide to somatic synthetic seed, what might be the different advantages or disadvantages, you can study it by making the synthetic seed. As well as we can able to know about what about the role of this testa and tegmen, that is seed coat. So how it will be observed in case of this synthetic seed, that can be also studied. Okay. Next one is also we can study the somaclonal variation. What is the meaning of that somaclonal variation? As because you are taking the callus, you cannot predict that this callus is of similar genotypes. Let us say you have taken a plant parts, okay? You can go for the callus production. So a callus, as the name indicates, callus are undifferentiated mass of tissue. So let us say you have taken the callus cells from here, okay? So let us say this one original plant 2n is equal to 2n is equal to, let us say, 24. So all these cells, whether all of them are 2n is equal to 24, yes or no. If it is, let us say, 2n is equal to, let us say, 26, this one is 2n is equal to 28, that means they are the variations from that parent parts. This explant is 2 and 24, here 26, 28, that means they show the variation. So if you have taken the callus from which embryo is being generated, later on plants is being generated, and if there is variation occur, then you can observe this somaclonal variation. So to know about somaclonal variation, this is also one of the methods. Okay. Also, we can take certain endangered species, okay, where it is going to be extinct from this nature, from this world. So to protect them, we can go for this synthetic seed technology. So more of these plants can be regenerated. Okay. Genetically modified plants or crops can be propagated. In some cases, what would happen? These GMO plants may or may not produce more of these seeds. So in that case, you can go for this artificial seed technology. You must have seen that certain fruits, if you are cut down that inside it, there will be no seeds. The best example is papaya. Okay. In that case, if you cut down, no seeds were there. So how can it will multiply? So for that reason, it requires synthetic seed technology. As well as some of the cereals part, fruits, medicinal plant, they can also be considered for this. In some cases, synthetic seed plantation can be done by using sowing farm machinery. Some of the farm machinery were there, which can be easily sowing the seeds that can be done by this one. Okay. And naturally, the main target of it, this one is more of the food production, and that too environmental friendly plantation. So more of the plantation can be done. So what might be the advantages? Very easily handling this one. You can easily store it. You can transport it. As I have shown a jar of that one. Okay. So transplantation, plantation can be also be easy. Expensive transport. Okay. So behind in small size, you can transport it to a large space. Storage life, it is much longer. Seed viability remains good for longer time period. Mostly longer means at times it will be 6 months. Okay. Product uniformity, you can able to produce the uniform somatic embryos because you have chosen it from the plant structure. Okay. Large scale propagation can also be done. This is also one of the plus point. Okay. And in some cases, mixed genotype can also be possible. Mostly 2 or 3 you can taken together. Okay. And also the rooting part hardening is very necessary over here. That will be can also be done over here. And transportation do not contain any disease causing agent because you have utilized different adjuvants. Okay. Because you have utilized different adjuvants, so there is no question of any type of disease causing agents. But a little bit disadvantage. As I mentioned earlier, this is very cost effective, but however, it cannot be used for soil transfer as they show very poor germination efficiency. For that reason, you have to go for initially the polythene bags, try to germinate over here. After attaining certain germination, then you can go for the soil transfer. Directly, you cannot switch over to this one. Normally, seeds, the normal seed you can pour in the soil condition and it can be easily grown for that reason, but it cannot be directly showing in the soil. Okay, and you just mark here the efficiency of, in case of particular lettuce plant, it is very less, that is 2%. Okay, is the only plant which was tried in the field. So directly if you apply into the field, the efficiency will be very less, around 2%. To avoid from it, you better go for the hardening stages. Okay, so these are the disadvantages. So I hope it will be clear to you. If some late entry has been done, let me first initially show this slide so it will be easy to understand. Okay, this is the artificial seed definition. So if you have any query, you can ask.
Hello, student. Please, if you have any query or the question or any doubt, please ask to the sir. Hello.
No, sir.
Actually, whatever parts I have mentioned where question may appear, I have already mentioned. You just go through it, and whatever bold part I have mentioned, those were may able to ask in your exam. So be ready.